Okay, so we're going to do a demo now, and the demo that we've chosen to show you is succinate production. So succinate, you might have heard of. It's a pretty important metabolite in central metabolism, and it's also being made by a couple of companies in the synthetic biology industry. One of them is Lygos, the other one is Miriam, and it's used as a precursor chemical for a whole number of things, including things like paint thinners. So it's a pretty important chemical that currently is being made by a petrochemical industry, and the synthetic biology industry is picked this up and is converting sugar into succinate. So talk us through a workflow that a company might use for the production of succinate. And maybe you can just pull up Riffyn and just show us how you start off. Maybe grab something and throw it into this framework that you're seeing. So if I wanted to start from scratch and just pull something up and throw in something, how would I do that?
Yeah, so here's a process that's partially made.
Okay.
Right. So we've got inoculate media, pellet and wash cells, and then batch fermentation. So if we want to say, okay, what do I do after this? Well, maybe I'll take some samples. So I can just add a step here and say, okay, this is my take sample step. Oops, if I could spell. Here we go. And then we can say, okay, what do we do after sampling? Well, maybe we're going to do metabolite analysis.
Got it. So you can build up this visual pathway of your experiment. And so go back to the one that you made earlier. So that's the whole thing that you've got here. So you've got inoculate media. So you've got engineered strains that you've made. They all produce succinate. You want to find out how can we maximize the yield, and what are the variables that affect that yield production?
Yeah, that's right. So what we're going to do in Riffyn is transform their protocol into a visual platform for experimental design.
Okay.
So you'll see here that here is their procedure represented as all these different steps.
Okay.
And so you see it visually. There's also a platform for recording videos. So here's a video of doing sterile inoculation.
And that might be if you've got a difficult protocol and somebody new joins the lab and you don't want to have to show them every time, so you can just take a video on your iPhone and upload it to
That's right. We've gotten feedback that this is a very useful tool, especially as there's turnover.
Right.
You don't have to keep retraining people. You can just say, oh, just go check out the Riffyn process. There's videos there.
Got it. So in this example, again, we've got different strains of yeast in this example. We're pelleting them, we're washing them, we're putting them inside a fermenter, inside a bioreactor, and then we're going to take samples over time. And then we're going to look at the output of these experiments. And so you've got three different outputs there. So just talk us through what those are.
Yeah, so this is measuring the ODs, that's the optical density. So that's for you to measure the growth rate.
Okay.
This is HPLC analysis, where you're going to look at the metabolite profile.
Okay.
And this is the biomass dry weight analysis. So that's where you're putting the cells on a filter and drying it, and then you're looking at biomass production.
Got it. So HPLC, high performance liquid chromatography. So you're actually looking at the succinate and how much of it is produced.
Yes, that's right.
Molecular analysis.
That's right.
Okay, great. So now show us an example of the old way of doing this. You had some data that you showed me before. If you can pull that up. Okay, so I'm very familiar with this. This looks like just my PhD thesis and what I would do in graduate school. So you've got 4 strains there, and you've got a few different conditions that you're growing these strains under. And you can't quite see, or you can't see on this monitor, but the strain 1 is orangey and strain 4 is red. It's a little bit of the same color. So you've got and then the circles are the media, the different media, and the triangles are the different pH. So you're trying to find out, your goal, if you so choose to accept it, is to find out which of these strains performs better under which condition. So, and this is what I do in graduate school. I kind of put my finger into the air and I pick one and think that maybe it's a little bit more than the other, and I choose that, or I choose a parameter to then optimize or something else to go after. That's the old way of doing things.
Yep, that's right. Yeah, so here, this is typical of how many of our customers are analyzing their data today. So they just plot it, and then they colored by strain, and they said, okay, I varied strain and I varied pH, and I want to understand the contribution of those two. And so what they see here is that the circles and the triangles are on top of each other, indicating that no effect of pH. But there appears to be an effect of strain, right? And the orange, which you're right doesn't look so hot here, it looks orange on my screen, is that looks like like the best strain, so that's strain 4.
Okay.
But what I'm going to show you today in my case study is that strain 4 is actually not the best strain. It's actually strain 2.
Okay.
And you would never know this just by plotting the data, as many of our customers are doing.
Got it. So go back to now the big plot. So you've now run this, you've entered all of your data and all of your parameters into this workflow, and then you've got the data, the data that comes out. So now, can you show me the machine learning? Where does that come into play?
Yeah. Okay. So what we're going to do at Riffyn is automatically annotate and automatically join your data.
Okay.
The moment that you collect it. So let me show you how we do that.
Okay. Great.
And so it all starts with a process flow diagram, as we've already walked through, and then the next step here is to define your inputs and outputs for those steps. So here we've got cell bank and media as the input to inoculate media, which creates a seed culture. The seed culture gets transformed, or it gets passed through after washing into the batch fermentation step, which creates a cell culture that then gets sampled and sent to all these different analyses. So that's the material and sample flow across the steps. And then the third component here are the properties.
Okay.
So if we look down here, you'll see that, for instance, cell bank, which is an input to inoculate media, has a number of properties like lot number, volume, strain name, and these are placeholders for your data.
Okay.
So what you're putting together here is a reusable template for recording experimental data. So you're saying that when you record data on experiments, you want to record lot number, volume, name, etc. And so what you've done here is really created a data collection engine, because what looks to you and I like a process flow diagram that I might draw on Visio is actually telling Riffyn on the back end to join all the data. So when the seed culture, which is the output of step 1, becomes the input to step 2, Riffyn knows to join that data.
Got it.
And then when this output becomes the input here, we join the data. So let me show you what that looks like in an actual example. So as I've stated, this is the template for data analysis, and template for data collection. Here's an actual experiment where we recorded data on an experiment. So just to orient you, you saw before that those properties were going down the screen, and here in experiment mode, they're going across the screen. So here we've got 2 strains that were being used to inoculate media, just to show you the flow of data here. So that one strain is now being split and to inoculate 2 bioreactors at 2 different pHs. And then if I just click on between batch fermentation and HPLC analysis, you'll see that the strain 1 is now being flowed into 15 samples are taken for strain 1, and then you'll see 15 samples for strain 2, etc. And so this, to me, is a really good interface for visualizing how the data are connected. But let me show you what it looked like before using Riffyn. So if we come here, I'll show you how our users are typically recording their data. So this is a batch record, and so this contains a lot of procedural information about how they're running their experiment. But what you'll notice here is that there's a lot of data here.
Okay.
So like the lot numbers of the strains, the lot numbers of the media, there's pHs, shaker speeds, etc. All of these are variables that could affect my key outcome. So my key outcome here, remember, is succinate yield. Any of these could affect the yield, but the way that they're being recorded now is a non-structured, non-mineable format. So if later on I want to try to correlate, say, lot number of my strain with the yield of my succinate, there's really no easy way to do it.
So go back to Riffyn and just show me when then the data comes out of it. Show me in particular how that gets converted into something that you can intuitively use then.
Yeah, exactly. So once I have all of my data in, all I need to do is just come down here and say export data from this experiment.
Okay.
Or if I want to join all the data across all the experiments on the process, which is most likely what you want to do because you want to look at the data in aggregate, you can just come here and say export data from all of the experiments.
Got it.
So I'm just going to do that right now. So I'm going to download this Riffyn generated file. And I'm going to open this. I'm going to open it in JMP. So Riffyn generates a CSV file that you can open with any program. So I'm opening it with JMP. You could open it with Spotfire, with R, with Python. I downloaded it directly from our user interface, but you can also pull it out programmatically via the API.
So what Riffyn's allowed you to do is to really look at everything visually, collect all the data, tag it, annotate it, collect all the instrument data, all the strain data, all the metadata associated with it in one place, and you wouldn't have been able to do that before.
Yes, that's right. So after you put the data in Riffyn, it'll get automatically joined. Remember, let me just take a step back. Remember via these connections between the steps?
Yeah.
So if I go here, when I put in my batch fermentation data, and when I put in my HPLC metabolite data, it's going to get automatically joined to all the relevant data upstream and downstream.
Got
so
it.
that the end product looks like this.
Got it. So now we're back into Jump, and now we can do the analysis. So yeah.
So just to orient you here, so this this data table you'll recognize is all the steps of my process.
Got it.
And then if I expand this, you'll see that all of those properties that you saw in the process are now in the data table.
Got it.
So all that, all the metadata, all the procedural data I talked about earlier, the lot numbers, the pH, the temperature, the fermentation data, the HPLC data, all here.
So show me now which ones correlate and which ones don't. How do I find that?
Yeah, so let's do that. So we'll do our correlation analysis. So this is back to that same plot, right?
Okay.
We talked about before.
This is the old way of doing
This is the old way of doing things.
Okay.
But when you have a Riffyn generated data table, it allows you to do these correlation analyses.
Got it. So I'm seeing your succinate yield there versus the theoretical yield. Okay, that's showing that the parameters are aligned. What have we got over there?
Yeah, so what this plot is telling us is that the variables in our model are accurately predicting the succinate yield, and the
Okay.
variables in our model are strain, pH, and temperature.
Got it.
So
we
you'll
can see a very strong correlation there. No correlation there. So this one is
This is the strain. So there's an effective strain.
versus yield. This is pH versus yield, so no correlation with pH.
Right.
And then this one is temperature, so strong correlation with temperature.
Right.
Okay.
And so if you noticed before when we were just plotting, the only two variables that they purposely varied were strain and pH. And so what looked like before an effective strain was actually confounded by the fact that it was an effective strain and temperature. So temperature they didn't mean to vary, but maybe operator error or drift in their instrumentation, they actually performed these fermentations across different temperatures.
Got it.
And so now they're seeing that actually there's a large effect of temperature. So when you correct for that effect of temperature, this is the actual order of the strains in terms of their succinate yield.
Got
So
it.
strain 2 turns out to be the best. If we go back to this plot, strain 2 is in green here. It looks like it's worse than 4, but that was only because these were run at different temperatures. So when
Got
I
it.
take that into account, now I can accurately identify the top producing strain and move that into scale up versus the mediocre one that I would have chosen before.
Got it. So if you can just go back to the Riffyn screen again. So again, what you're seeing is a visual workflow, and you can see here all the different steps of the protocol here. You can see the data is annotated, the metadata is attached to it, and then the data that it spits out the other end, you can then run these statistical machine learning algorithms on it. And there's basically hidden, the secret, the answer of which strain to use was hidden inside this data. A human brain couldn't analyze it. You can't see it by a visual analysis. The machine learning can, and it pulls out and tells you which parameters to optimize and how to increase your yield, and it tells you which experiment to do next.
Yeah, that's right. So one of our customers prior to using Riffyn, they said, we work on what we know, not necessarily what matters. And with Riffyn, we allow you to flip that around, and we allow you to work on what matters, not necessarily what you knew going into the experiment. Because once you do the analysis, you didn't know that temperature had such a large effect. Now we know it does. So now, moving to scale up, we know, hey, we need to control for temperature. And in fact, this looks like a good variable for us to vary to try and increase the yield of our strains.
Got it. Excellent. Laura, thank you so much.